Oh, I see that Sergio has already put his slides up. Wonderful.
I thought we should be
You,
efficient.
uh-huh. It's been my pleasure to meet and work with Sergio for, gosh, how many years? 2 or 3? Well, actually, even earlier than that, because it was the lab that Sergio was working in that extracted the skin cells of 2 of our Timothy syndrome patients that you did the IPS cell studies on. From those studies in that laboratory, they found out that developmentally the Timothy syndrome myocardial and neuronal cells developed abnormally. And then just about 3 years ago, Sergio actually discovered with his group that these neurons do not migrate properly. So I'm really excited to hear what he and his associate, Dr. Rebecca Levy, has to say. I think the moms are going to be blown away once again with all that you're going to share with them.
Well, thank you so much, Catherine, and thank you, everyone. We're very excited to both me and Becky today to share with you some of the work that we've been doing in understanding the neuronal abnormalities in Timothy syndrome, but also very excited because Becky is embarking on a very interesting project now, which she will mention. And she actually just a couple of weeks ago got a grant that will actually support this type of research, which is very exciting. There are not many grants for Timothy syndrome, so I think this is a very important reason to celebrate. I'm a physician by training. I actually don't see patients anymore, but my interest has always been in understanding the biology of neurodevelopmental disorders, in particular autism spectrum disorders. And I can tell you right from the start that one of the major challenges that we have in understanding all of these neuropsychiatric disorders is actually our lack of access to the developing human brain. And by lack of access to the developing human brain, I really mean lack of access at the molecular and cellular level. We may be able to image the brains of patients. Well, in the case of Timothy syndrome, as we've heard, there are actually no studies published so far. But even those studies would not really give us the resolution that we really need in order to understand the biology of these disorders. And one of the challenges with studying the human brain in general is just simply how long it takes to develop. So this is just a very simple schematic that shows the development of the human brain from very early stages, 23 days, as you can see here, all the way into adulthood. And you see that actually even at birth, the brain is still not fully developed. It takes, just to give you a sense, it takes about 25 to 26 weeks to make all the neurons in the cerebral cortex. And when neurons are already made in the cerebral cortex, the job of the brain to really function is not yet done because you still need glial cells of various subtypes. And for instance, myelination, which was brought several times today, really only starts postnatally and continues in humans up to the second and third decade of life. And so this inaccessibility at the molecular and cellular level to tissue, I think is one of the major challenges that we've been facing and we continue to face in trying to understand the molecular biology of these disorders. And that's why I often joke that I suffer from what I call an oncology envy syndrome, which is of course not a real syndrome. It's a made up syndrome, but it essentially illustrates the frustration that we very often feel in psychiatry and in neurology when we see just how fast cancer research has moved in the last 3 or 4 decades, and how slow we've been moving in psychiatry and neurology in developing new therapeutics. And of course, there are a lot of reasons for why cancer research has moved faster, but I think one of the major reasons has been our inability to directly access the human brain, which, as you can imagine, for cancer researchers is much easier because most of those tumors are removed or can be studied following a biopsy. And so towards this, we've been working, I guess now almost for 12 years, in trying to build in a non-invasive way neurons from patients that we can study directly in a dish. And I think there's no better way of illustrating this approach than through the eyes of a patient. So one of my first patients sent me this drawing when I started my lab. And he's a high functioning patient with autism today, actually a quite accomplished composer. And he thought, and I'm going to paraphrase him, he thought that what we were doing is that we were climbing up on a ladder and then poking holes in people's brains and using telescopes to look at neurons and glial cells. And as you can imagine, this is not what we're doing. All right, so I had a conversation with him and I explained, look, this is like what we try to do, but this is not how we do it. And I explained to him like how we're actually approaching this experimentally. And next morning, he sent me another drawing, which I think it's a pretty accurate representation of the experiments in my lab. And again, to paraphrase him, what he said that you're doing is you're taking skin cells from these patients and then doing some mumbo jumbo to the cells to turn them back in stem cells. And then he knew that stem cells can become any other cell type in the human body. So he knew that we were trying to guide them to become neurons and glial cells in a dish. And really, this has been the approach that we've been using. And I'm really, in a way, very proud because I moved into this field when the cell reprogramming was discovered. And in fact, we made some of the first neurons that were ever developed through these technologies were actually done from Timothy syndrome patients. And so a number of years ago, I guess it's almost, well, when we published this paper, it's almost 9 years now. But what we essentially did is we recruited a small cohort of patients at that time with Catherine's help. At that time, 2, we now have actually 3 patients and some atypical as well. But all of the patients at that time had the classical mutation, the G406R mutation. And then essentially what we did is we took skin cells from these patients through a biopsy. We reprogrammed the cells by pushing them in time to become stem cells. And then we guided those stem cells to become neurons. And you can see, for instance, here's one of these neurons from a patient with Timothy syndrome that has been actually derived in a dish. And you can see as it kind of like extends and retracts some of its processes. And again, this is completely non-invasively, it does not involve anything else but a skin biopsy. And we've actually spent a lot of time studying this over the years. And what I want to do in the next 5 or 6 minutes is show you some of the abnormalities that we've been identifying. Now, I want to remind you that even at that time, when we did this experiment, well, it was thought that this mutation was causing a gain of function, so that more calcium would get through this channel. That was not really proven in human neurons. And so the first experiment that we've done is actually to take these neurons and measure calcium inside the cells. They're excitable cells. So we would excite the cells and look at calcium going up and down in the cells. You can see here by the green color, at one point they get depolarized, which happens like now, you'll see in a second now, right? Then you see calcium goes up and then it goes down. And this very simple experiment showed us here, as you can see from this graph, that when you depolarize control cells in blue, calcium goes up and then it comes back. But when you do this in patient cells, calcium goes up, but it fails to come down at the same rate. And if you block that calcium channel with one of the drugs that I was mentioning before in the conversation called nimodipine, you can actually completely stop that defect, which again shows us that it is mediated through those channels. So in a way, this was expected, but it was a much needed experiment to prove that those neurons really have this calcium abnormality. Now, a colleague of mine, Masayuki Yazawa, has his own lab now at Columbia, also did similar experiments where he actually derived cardiomyocytes and aggregated them in such a way that they actually beat. So you can see, for instance, control cardiomyocytes put all together as they're beating in a dish. They beat at about 60 beats per minute. But then you can see, for instance, Timothy syndrome derived cardiomyocytes, which I think is very visible from this movie as well, that they don't beat regularly. They don't beat at the same pace. And Masayuki actually spent a lot of time and did this also in his own lab afterwards, trying to characterize what are the cellular molecular mechanisms underlying this defective contraction process. But it was not just that there was an abnormal calcium in the cells. We found that one of the earliest defects that is present in the cells is in the way the cells expand their processes. And so if you take control neurons and you start exciting them, depolarizing them, they will tend to extend their dendrites, their neurites, just essentially just become larger. And you can see this here happening for control cells over about 2 hours of doing this. But when you take Timothy syndrome neurons derived, Timothy syndrome neurons that are derived from patients, you can notice here that instead of extending their dendrites, actually they retract their dendrites. It turns out that this defect is actually not mediated by calcium. It's interestingly independent of calcium. So it also means that some of these changes are dependent on calcium and others are not. So in this case, we think it's actually a conformational change of the calcium channel that triggers a signaling cascade downstream. We also found an interesting phenotype that we're still pursuing now and became even more interesting in recent years, because we found that neurons from patients were ectopically expressing an enzyme that is important for producing neurotransmitters such as dopamine and norepinephrine. And initially, we were a little bit puzzled because cortical neurons generally, the neurons in the cortex don't usually produce these enzymes. But it turns out, and this really was just like proven independently by Nenad Sestan's group at Yale, is that in humans, specifically in certain type of primates, not just in humans, these cells are an evolutionary innovation. And it's unknown exactly what they're doing, but it is thought that this release locally, this neuromodulatory processes locally in the cortex may be underlying some of the evolutionary changes across primates. And we've shown that they can actually produce norepinephrine and dopamine. We've also found that, and this was a little bit surprising to us at that time and confirmed that afterwards, that the type of neurons that are generated in the cortex is actually different in patients and in controls. So it's not just that the cells don't handle calcium really well, but in fact that as the cells are being generated, different types of cells are generated in the cortex. In a way, there is a bias for certain cell types versus others. In particular, we found that there was a bias in the upper versus deep layer neurons. And we've shown this initially in kind of like almost like early experiments in human neurons. But Georgia Panagiotakos, who also now has her own lab at UCSF, took this really to the next level to prove that indeed this channel, actually the way the channel is spliced is essential for the generation of various types of cortical neurons. And this paper was actually published just earlier this year, showing that by playing with the levels of the various type of spliceoforms, Georgia could actually change the type of neurons that were generated in the cortex. And here is one thing which I don't know how many of you know or are aware of, because this is also in the context of the splicing discussion before. The mutation that causes the type 1 Timothy syndrome is in one of the splicing variants. It's what we call the minor splicing variant. It turns out that minor splicing variant is the major type of calcium channel present in early development. So we think that because that channel is present very early, abnormal channel is actually causing these abnormalities. And that's something that we're still discussing. But of course, there were a number of challenges with some of these cultures that we were developing in the beginning. Most of them are kind of at the bottom of a dish, as you can see them here. They are neither very complex in terms of their cell diversity. You can't really model many processes, and you can't even look at maturation over long periods of time. So in the last 6 or 7 years, my lab has been developing a completely new set of tools and approaches to develop 3-dimensional brain tissue from patients that allows us to study even more complex cellular processes. And so these cultures are now known by many names. They're initially, because they're very spherical, we call them spheroids, but they're also known as brain organoids, or more specifically, the ones that we've developed are known as brain region specific organoids. And essentially the process is the same. You take fibroblasts from patients, you reprogram them into pluripotent stem cells, and then you aggregate them in essentially balls of cells that we later differentiate over long periods of time and essentially just stay floating for very long periods of time. And we've shown that we can, just to get a sense of like how these cultures look like, this is a mouse brain during development on the left, and this is one of our brain organoids on the right. So as you can see, they can grow pretty large in size, up to about 4 millimeters. But the advantage of this is that they're derived non-invasively, and we can derive essentially thousands of these cultures in a dish. Because they're three-dimensional, they have a beautiful architecture inside. They have a ventricle-like region inside. We've shown that they make multiple neuronal layers over time. You can look actually at calcium signaling right inside this culture, so you can see how they're flickering and essentially trying to talk to each other. And if you wait for longer, you can get both astrocytes as well as oligodendrocytes and myelinating oligodendrocytes. You can also look at how cells are talking to each other or interacting with each other. So you can see, for instance, how neurons and glial cells, neurons are shown here in red and glial cells are shown in green, how they're actually interacting with each other. Here, we just plated them on top of a glass cover slip. And the moment we plate them down, as you can see, the green cells, which are the astrocytes, the ones that usually respond to injury in the brain, start expanding their processes towards that surface to just see what is happening. Neurons don't care about that. But neurons do care about finding other cells. So you see there's a lot of dynamic changes here. You'll see right now, this is a neuron, this is a human neuron expanding these processes here, trying to find other neurons. There are none around it, but that's what they try to do. And these are actually derived from patients that have a specific form of schizophrenia. And there's not necessarily something abnormal here, but I think this illustrates the power of this technology that allows us within months to just derive these dynamical cultures that contain both neurons and astrocytes from patients in a non-invasive way. So initially, we've been just deriving specific brain regions, and I think this is highly relevant to Timothy syndrome as well. A number of years ago, as Catherine was mentioning, we developed a completely new approach that allows us to look at how brain regions talk to each other in a dish as well. And this essentially involves the same process, except that instead of making one brain region, we use various cocktails of growth factors and small molecules to derive other brain regions separately. And once we have derived them separately, we essentially put them together at the bottom of a tube, and we allow them to fuse with each other in a preparation that is now known as an assembloid. And essentially, this is very simple and very straightforward. This was published a few years ago, but essentially you derive them separately. This tiny little structures here resemble different parts of the developing brain. You make them separately, and then you put them in a dish, and then within 48 hours or so, if you put them in contact with each other, they have fused. Why do we want to do this? Because a lot of the action in the brain is in this interaction between brain regions. So for instance, if you put them together, these two domains of the forebrain, the dorsal and the ventral forebrain, initially nothing really happens. But if you wait for about 3 weeks or so, you see that most of the green cells that were here in brain region number 2 have now actually moved towards the other brain regions. These are neurons called GABAergic cells or interneurons. They're about 20% of the neurons in our cerebral cortex, and they're absolutely essential for the function of the cortex because these are the ones that put the brake on electrical activity in the cortex. These are very important cells for instance, for epilepsy. And they're also thought that how they interact with glutamatergic cells are also important for autism. And so we've shown that once we put the cells together, we can literally watch them as they move from one side to the other side and watch this migration very carefully. This is the reason why this was so exciting also at that time, is that this type of migration has not really been observed in humans. This type of migration happens in the 2nd and 3rd trimester. So it's been very, very challenging to watch it live. And this brings us to Timothy syndrome because we've actually investigated this in Timothy syndrome. There was an earlier report from Frank Polleu's lab who showed in mice that if you block L-calcium channels in these migrating cells, you can essentially completely block them from moving. So we thought, well, now if we have patients that carry this mutation, that is a gain of function mutation, what would happen to these interneurons? If they have more calcium going inside the cells, will they move faster or will they move slower? And the answer is both. Because what we actually found is that Timothy syndrome derived GABAergic cells tend to engage in this migratory movements much more often, but every single time they move a shorter distance. So when you watch them over time, they're essentially left behind. Now, known by using various drugs that if we apply drugs that block this calcium channel, you can, as you would expect, completely block the migration of interneurons, but actually you can restore the migration behavior to normal levels of Timothy syndrome neurons. This shows that you can actually pharmacologically intervene and manipulate the cells. And I can go into a lot of other details. Fikri Virey in the lab has actually been spending a lot of time now to try to really carefully understand the molecular mechanism behind this and find better way of restoring this. This paper is very soon gonna be ready, and I'll be happy to share with you like more details. This is a very exciting story that is coming together. But in general, one of the things that we've been doing, talking about other brain regions, is trying to make various brain regions and assemble them 2 by 2 or 3 by 3 to show how they're interacting. And we're going to have a paper that is about to come out hopefully in a few weeks, where we've actually assembled one of the most complex circuits today in a dish. We've assembled the corticospinal muscle circuit, which I think is also relevant to some of the discussions that we have here today. And I'm not going to go into all the details, but essentially what we did is we derived a spinal cord from the stem cells in a dish. We derived a cortex, and we derived muscle cells. And then we essentially put all three together, all made from stem cells separately, and then put together. You can see the cortex here, the spinal cord in the middle, and the muscle. You see the muscle fibers here. And this can be derived from any patient, from any subject. And what we've shown is that when we put them together, very interestingly, the muscle actually starts to contract. And it starts to contract usually when we stimulate the cortex. So if you electrically stimulate the cortex, either with light or with glutamate uncaging and other methods, you get this beautiful, reliable contraction of the muscle. So this gives us a very powerful preparation to start looking at more complex circuits in a dish. And I think this is going to be highly relevant, I think, for Timothy syndrome as well, especially in the context of the discussions that we've been having before about the expression of this channel. And with this, I want to wrap up the basic science part that we've been doing. And this is my lab before COVID. And this is my lab actually yesterday, where we had one of our first like get-together, safe get-together on the Stanford campus. Of course, everybody's wearing masks, and we celebrated with some individually wrapped cupcakes, a few papers, and a few new people joining the lab. And with this, I want to thank you, and I want to let Becky tell you about the exciting projects that she has up and running. I think you're muted, Becky.
Is that better?
Yes.
Okay, thank you so much, Sergiu. And it's so nice to meet everyone virtually. And like everyone else, I'm so sad that we're not all together in order to be able to meet, but I hope everyone's safe and healthy. And I wanted to share with you folks a project that we were hoping to start in order to try to expand the neurodevelopmental symptoms that we are learning about in Timothy syndrome. And as was mentioned earlier, there's a difference between what all of you as families know about what your children's symptoms, strengths, concerns have been, and what we're able to see as researchers out there in the literature. And sometimes filling in that gap really helps us make big strides forward. And so if someone goes and they looks in the literature, here's some of the fantastic papers that have already been out there telling researchers and clinicians, as they're starting to learn about Timothy syndrome, what's already known. So it's known that, for instance, there's a risk of seizures, that there's some children who have been tested and are on the autism spectrum. Some children have intellectual disability or just developmental delay. And some children have hypotonia, in other words, that their bodies are a little bit looser. And also some patients have some mood symptoms. And it's not totally clear to us how that changes in the different types of Timothy syndrome. But we're hoping that we could fill in a few of these gaps. As Dr. Tunbridge kind of referenced a little bit of this, and I know it's been spoken about throughout the day, when we look all across populations, we know that the CACNA1C gene is associated with autism, with bipolar, with a disorder, with depression and schizophrenia. But we really think that it's time that we try to better understand what are our patients' strengths and what are their challenges. And that will help us direct therapeutic efforts towards what it is that will most help patients. And we hope that as we do this, we'll get insight into the pathways affected in these more genetically complex neurodevelopmental disorders. And so we proposed to do a study, and fortunately we just got a little bit of grant funding and also just had our IRB reapproved yesterday, which has been exciting. We know that all of you have an amazing amount of experience with Timothy syndrome in what you've seen either as a clinician or a researcher or as a family. And using some of that, we've put together a survey that we would like to distribute if people are interested. It could either be mailed or it could be done online. I can follow up with anyone who's interested after they fill out the survey, kind of follow up any questions or concerns that you feel weren't covered inside of there, because we're really curious if you have other thoughts. Then we're interested if families are interested, and it's optional because this was kind of put together before the coronavirus pandemic, but we're interested in being able to do a blood draw in order to confirm the genotype for Timothy syndrome. And also, if families are interested, to consider banking blood in order to think about making some of the induced pluripotent stem cell lines that Sergio was just talking about in order to think about new neurospecific tissue lines. And we hope that this will turn back, as Sergio was just telling us, that there's all this really exciting research going on the electrical and physical properties in neural development. And in turn, the more we learn from doing that, the more we know about how to diagnose and treat and help all of the patients with Timothy syndrome. So if you're interested in the survey, here's my email address. You can also reach me through Stanford or get in contact with Catherine. Again, the blood draw is something that we're trying to figure out a way for that to be done safely if families wish during coronavirus. And we just want to give so many thanks to Catherine, to everyone at the SADS Foundation and the TSA, and also to John Bernstein and Elijah Kravitz, who have been helping us get this clinical study off the ground. So with that, I'll pause so we can answer some questions.
Thank you so